Hello student. Today we are going to discuss the topic inheritance and variation. So in this topic we are going to study concern with the different points. Today in short we are going to study chromosomal theory of inheritance, structure of chromosome and the types of chromosome. But before starting the actual topic, you have to know what is heredity, what is variation. So heredity can be defined as the inheritance or the transmission of characters from one generation to the next generation. Whereas the difference existing between parents and their offspring, it is going to be known as variation. The scientific study of mechanism of heredity as well as the causes of variation, it is nothing but genetics. The Mendel is going to be known as the father of genetics. It is very well known to you that the living organisms are made up of cells. Cells may be prokaryotic cell, eukaryotic cell. The cells with the nucleus are eukaryotic cells, which show the typical chromosome, whereas the cells without nucleus are prokaryotic cells, which show the irregularly folded mass of genetic material known as nucleoid. In case of prokaryotes, the chromosome is single and made up of circular DNA, which is also going to be known as plasmid, and it is capable of independent existence. Now, in the next slide, we are going to study about the chromosomal theory of inheritance. So, this theory was put forth by the Sutton and Boehry in 1904. This is the theory of genetics which help us to identify that the chromosomes as the carrier of genetic material. So this chromosomal theory of inheritance states that in short, the gametes carry all the hereditary characters. They are linked between parents as well as offspring. The nucleus of gametes contains the chromosome which carry all hereditary characters. The chromosomes are found in pairs in somatic or the diploid cell. But when the gamete formation takes place, the segregation or the separation takes place and each gamete gets formed which carry only one set of chromosomes. During the fertilization, due to the union of gametes, the restoration of the diploid number of chromosomes of that species, it is taking place. So it is very well clear that the chromosomes acts as a carrier for the genetic material, or the chromosomes are responsible to inherit the certain characters from one generation to the next generation, or from parents to their offspring. In the next slide, we are going to study concern with the chromosomes. So what are that chromosomes? Chromosomes are the filamentous bodies present in the nucleus of the eukaryotic cell. These chromosomes are visible only during the cell division. The meaning of chromosome, it is nothing but the chroma means color and the soma, it is nothing but body. That is nothing but the color body. It is nothing but chromosome. So chromosomes have the ability of their self-replication and they play the important role in heredity, in variation, in mutation as well as in evolution of characters. The chromosomes which are structurally as well as morphologically identical are going to be known as homologous chromosome. Generally, we are going to find it out. The number of chromosome, it is specific or it is constant for a particular species. The presence of whole set of chromosome Before that, you have to know what is polyploidy. Polyploidy, it is nothing but the repetition of primary basic number of chromosomes, which basically divides into the two types. That is nothing but euploidy, and the second, it is nothing but aneuploidy. So euploidy, it is nothing but the presence of the whole set of chromosomes, right, which further divides into the different two types or into the different three types, which are nothing but monoploidy, diploidy, as well as polyploidy. The polyploidy, it divides into the two basic type, autopolyploidy as well as allopolyploidy. The second type of the polyploidy, it is nothing but aneuploidy. It is nothing but either addition or either deletion of one or more chromosomes to the total number of chromosomes which are present in the cell. So this aneuploidy, it further divides into the two types, which are nothing but hypopolyploidy and hyperpolyploidy. The hypopolyploidy further divides into the two types, monosomy as well as nucleosomy. Hypo means what? The deletion of chromosome. Whereas the hyperpolyploidy, it divides into the different two types, trisomy as well as tetrasomy. Trisomy, it is nothing but addition of extra one chromosome, and tetrasomy, it is nothing but the addition of two more extra chromosomes. So in case of chromosome, generally we are going to find it out that the size of chromosome varies from species to species. The shape of the chromosome also varies according to the stages of cell division. In the next slide, we are going to study concern with the structure of chromosome. So generally, the metaphase chromosome has the two identical walls, which are nothing but, in short, we are going to consider as the sister chromatids. Each chromatid is term is made up of subchromatids, or which are also going to be known as chromatonema. So here you can see the chromatonema. In between the chromatids, the one point is present. That point, it is going to be known as the centromere. So this is the centromere. The centromere, it is also going to be known as the primary constriction. During the cell division, the spindle fibers are attached at the centromere. Besides the primary constriction, the additional narrow area, it is also present, which is also going to be known as the secondary constriction. So here you can see the secondary constriction, right? Some secondary constriction also known as the nucleolar organization, because they are necessary for the formation of nucleolus. The part of chromosome beyond the nucleolar organization, it is short and it is generally going to be known as the satellite. So this is nothing but the satellite that we are going to get. It is the satellite. So before that, also you are going to get the part that is nothing but the telomere. Telomere, it is also present, which is the unique property which prevents the ends of the chromosome from sticking together, but the attaches to the nuclear envelope. The surface of chromosome bears the number of small swelling. That swellings are going to be known as chromomeres. So this is in short the structure of chromosome, which shows the two sister chromatids, chromatonema, along with the centromere, primary constriction, secondary constriction, satellite, along with the telomere. The next slide that we are going to study concern with the sex chromosome. So two sex chromosomes are there, X chromosome as well as Y chromosome. So when you are going to see The structure of X chromosome, Y chromosome. The X chromosome it is longer, Y chromosome it is shorter. The X chromosome contains the large amount of euchromatin and the small amount of heterochromatin. So generally euchromatin contain the large amount of DNA or the genes, whereas the Y chromosome contains the small amount of euchromatin and a large amount of heterochromatin. So less amount of DNA. So genetically Y chromosome it is less active. Both X chromosome as well as Y chromosome show the two regions. That two regions are nothing but homologous region as well as non-homologous region. So it is the non-homologous region of X chromosome. It is the non-homologous region of Y chromosome. So non-homologous region of X chromosome, it is responsible to contains the gene which carry the X-linked genes, whereas the non-homologous region of Y chromosome contains the Y-linked gene, whereas the homologous region of X chromosome as well as the homologous region, the non-homologous region of Y chromosome, they show the dissimilar genes, and the crossing over it is generally occur in case of non-homologous region of X chromosome as well as Y chromosome. In the next slide, in short, we are going to study concern with the types of chromosome. So according to the position of centromere which is present, we are dividing the chromosome basically into the four types, such as the metacentric chromosome, submetacentric chromosome, acrocentric chromosome, and telocentric chromosome. In case of metacentric chromosome, the centromere it is present at the center and which has the two equal arms or the limbs. So that chromosomes are known as metacentric chromosome. In case of submetacentric chromosome, we are finding out the position of centromere. It is slightly away from the center position. So generally we are going to find it out. The one limb or the one arm it is shorter and the one arm it is longer. So that chromosome it is known as submetacentric chromosome. The third that we are going to get is the acrocentric chromosome, in which we are going to find it out the position of centromere. It is present towards the one end, right? So which has the very shortest one arm and very shortest or very longest one arm. So that chromosome are known as acrocentric chromosome. In case of fourth chromosome, that is the in case of telocentric chromosome, the centromere it is present towards the one fourth and which has only one arm. So that chromosome it is known as the telocentric chromosome. Generally, the metacentric chromosome appears V in shape. The submetacentric chromosome appears L in shape. Acrocentric chromosome appears J in shape. And the telocentric chromosome, it is rod-shaped chromosome. So the chromosomes that we have studied may be metacentric, submetacentric, acrocentric, telocentric. They are responsible for the inheritance of characters. They regulate the protein synthesis and also help in the cell division as well as cell growth. They control the cell metabolism by directing the synthesis of enzyme. Whereas the sex chromosomes that we have studied, X chromosome as well as Y chromosome, they are essential for the determination of the sex. The chromosome replicate to produce the exact copies of themselves, which are passed on to the daughter cell. So in this topic, in short, we have studied concern with the chromosomal theory of inheritance. What are chromosomes? The structure of chromosome, sex chromosome, as well as the types of